Carolyn Maziel from Cornell, who's going to talk on the population size.
Okay, I would like to thank the organizers, especially because I'm in a population genetics session, and I'm pretty new to this too. Before that, I did pure phylogeny. So this is a part of a project which was a bigger scan on 6 mammalian genomes, and this project was very bit data-driven, so I actually have a real data slide next. So we took, this is 6 mammalian genomes, and the species were, the species were human, chimp, macaque, mouse, rat, and dog. We took sequences from different databases, and normally you can't trust those things, so we apply a couple of filters. And one thing which is important for later on is this starts to be kind of a proper phylogeny for genome scans, because you have 6 species. But sometimes you're actually just interested in part of the tree, so for example, you're interested in the rodents. And if there's problems with the sequences in the other species, so for example, if there's a problem in the macaque species, you can just treat that as missing data. So actually sometimes we just allow to be a species treated as missing data, or you have truncated genes where actually the axon boundaries are not conserved. In total, we kind of investigated 17,000 genes, around 500,000 genes, and we are mostly interested in the patterns of positive selection. And if you do this, a very standard way to do it actually is measuring selection pressures by using phylogenetic models. And a couple of people already talked about codon models, so I'm not going to say much. I only wanted to remind you about the non-synonymous weight ratio and its interpretation. So if omega is smaller than 1, we're talking about purifying selection. If it's equal to 1, it's neutral evolution. If it's larger than 1, it's positive selection. So first thing I did was I concatenated all the 17,500 genes and calculated a branch-wise non-synonymous weight ratio. And what I would like to point out, so for example, for human and chimp, it looks very similar. But for example, for macaque already, it's more purifying. And this kind of observation gets more extreme if you go to mouse and rat. So mouse and rat, the rodents, are more purifying than the primates in general. So this is just like concatenating all the genes, some sort of an average idea of the selection pressures. We also did this per gene, on a per gene basis. So here I split up the tree and calculate per gene in omega for the primates and the rodents. What you can see is, on average, you again see more purifying selection. But for the primates, there is actually a significant shift towards less strong purifying selection. And that shift, we did a Mann-Whitney, that shift is actually significant. So this means like if for species which seem to have a larger population size, like rodents, purifying selection is more efficient. What happens for positive selection? So we did also a positive selection scan on all those genes, quite standard branch side model. And we tested on the phylogeny if there's positive selection on any of the branches, but we also tested specific internal and external branches of the phylogeny or clades. And in total, like out of the 17,500 genes, we found 544 to show a signature of positive selection. So what are the tests we actually did? So the one in the middle of the tree you've already seen, and this is always the number of positively selected genes out of the genes we tested. At the beginning, I sometimes said that some of the genes are, not all of the genes are always treated in all the tests, because, for example, if you actually if you kind of mask off the rodents because the quality of the rodents is bad, you can't really do a positive selection scan on the rodents. So it's always number of positively selected genes out of the numbers which have been tested. So for the any case, is there a positive selection on any of the branches? You find 400 genes, that's the majority of the genes. For lineage-specific genes under positive selection, it's actually quite small numbers. So only 10 on human, a little bit more on chimp, 18, hominids 7, and macaque 16. Then there's something you actually can see. The primates, it's 24 genes, and nearly over double for the rodent clade. So again, here you kind of get the impression positive selection is also more efficient because you get higher numbers. But this one can also be misleading. So for example, in the upper one, it could be again that because of larger numbers in the primates, you see less positive selection in the primates than in the rodents. But on the other hand, this rodent branch is really long. So you're going to see a lot of changes. And that means your statistical power actually to detect positive selection is stronger too. So in some way, this thing could also be slightly misleading because of different power to detect positive selection. And the other thing I wanted to say is, so if I kind of find positive selection for a gene on this branch, it doesn't mean there's no positive selection on that branch. It also, or in the any case, it doesn't mean it's really on all the branches. It could be just very strong on a couple of the branches, episodically, and you get such a strong signal that you think on any of the branches or on all of the branches you see positive selection. So one other thing we did was we just took the 544 genes where we thought, well, there's some sort of evidence for positive selection. And we implemented some sort of Bayesian selection model. And here, this has the advantage that you kind of can calculate expected numbers of positively selected genes for the branches. So I'm not going to go too much in the model, but what I would like to point out is the numbers in red. This is like positively selected genes on that branch. It's an expected number for it. So for human and chimp, it's fairly similar. So that's the numbers in red. They're mostly small. The numbers in blue is if there's expected numbers of positively selected genes on that branch, but could be on any of the other branches too. So it's some sort of a marginal expected count. So what you can see is human and chimp are chimp is a bit higher, but the intervals overlap, so they're quite similar. Then again, macaque has a higher marginal count, which means like, again, it looks like that positive selection is more efficient on macaque. And then, like, the last thing I want to point out is, so here, we actually did it for the clades, for rodent and primate. And again, if you compare the numbers, so that's about 89.5 for the rodents and 50.1 for the primates. You see more positively selected genes on this clade, on the rodent clade than on the primate clade. So this is something it's not completely unknown. Some other people have observed this. This is a positive selection scan. And this is like how most positive selection scans before looked. You have quite simple phylogeny. And these, the authors backward are, they found that there's more positive selection on chimpanzee than on human. And they kind of really emphasize this. And then somewhere in the text, they give an explanation. They say, like, this is explainable by the reduced efficiency of natural selection in humans because they have a smaller population size. So I went back to the chimp paper. The problem with those genome scans always, it's only one individual. And chimpanzees have a lot of subspecies, and I just wanted to figure out what chimp that actually was. So this chimp is called Clint, and Clint is a West African chimpanzee. And the genome paper says like the diversity in West African chimpanzees is similar to that seen in human populations. And that would mean it's actually the same population size. Or they think it's the same population size. And Clint is broadly consistent with West African origin. Okay, so this is kind of two different opinions, and I hope I did a quite long motivation to give you an impression that for this for studying selection, the population size is really interesting. So it's a long motivation, probably. So if you don't believe me, it's interesting. Probably believe Arndt von Hessler that population size is an interesting thing, quite sexy, and you should kind of put it into your models. And this is now what I'm trying to do. So this is on the species level. On the species level, actually, we think that all the substitutions you kind of see are already fixed. So we start off with a very fundamental result from Kimura from '69. It's the probability of the fixation of a mutation. And what goes inside is the effective population size. And because we look at mammals, it's actually 2n because they have 2 chromosomes. And also there's a selection coefficient. If selection coefficient is larger than 1, it's selectively favored. And if it's smaller than if it's smaller than 0, it's selectively disfavored. And for the neutral case, so if 0 is 1, you can actually just apply. Here you just take the limit from this one and apply L'Hopital's rule, and you see it's just simply the initial frequencies of a mutation. So from this one, you can calculate the rate of mutation. So the rate of mutation is just the product of the rate of the mutation, the mutation rate and the population size plus and the probability of fixation, which we just calculated. And you can also do the same thing for neutral mutations. So now we have 2 rates, one for neutral mutations and the other one for selected mutations. And then we just do now an interpretation. So we have both rates. And the non-synonymous rate ratio, there's also a bit the thinking that a synonymous change is neutral and a non-synonymous change is selected. So here I kind of do the same things. I take the 2 rates and get a new interpretation of omega. And this is just plugging the formulas in, and then also we substitute 2ns with the gamma. This idea was first introduced by this interpretation of omega by Bruno and Harper. Nielsen and al. Kind of were interested in distributions of selection coefficients and used this formula for this. And then very recently, Thorne et al. Actually, he does far more complicated things with this, relating this also to tertiary structure. So what we're now going to do is actually a bit simpler. But first, before we start applying this to data, I would like to point out a couple of advantages and disadvantages of this kind of interpretation. So the advantage is now we're actually looking at human, chimp, macaque, and there's the possibility of multiple substitutions at site. This is simply because there's a larger divergence times than normally on a population genetic level. And so this has the advantage we can actually take this into account because it's standard probabilistic model. The other thing is like you can, like omega, you can actually calculate it for each branch. And in some way, we kind of hope that this kind of improves the understanding how omega, the selection pressure, is affected by the population size. Disadvantage is this is more like things, I work in the population genetics lab now, which people tell me. The sites are assumed to be independent, and often in population genetics, it's not the case. The other thing is population geneticists are also a lot interested in this speciation event himself. So, they want to know whether there was some sort of isolation, there's migration between those things. You have island models for this. And this model is kind of really naive. It just assumes that at speciation there's kind of an instantaneous change in population size. So in some way, all the questions which are interesting for population geneticists are not really covered in this model. And the last problem, and that's probably one which is kind of normally a real bumper, is you actually just have this product. You don't not really have the selection coefficient or the population size separately. So normally this is kind of really not nice, but actually if you take ratios and if you have genes where you can assume that the selection coefficient is the same, you actually get at least ratios of population sizes. And this is what I've tried to do here. So I assumed that starting from the Nature paper, I kind of assumed that the population size of human chimp for that one chimp is the same, and the population size of macaque is different. But I now calculated like omega first, and then took the formula to calculate all the ratios, and I get a ratio between macaque and human of 1.4, and also have a confidence interval. So from variation data, people also calculated the effective population size. So for the ancestor of macaque, they get 73,000. For the ancestor of human, this is quite difficult to calculate. So people always give a pretty big range. So it's 40,000 to 70,000. And I kind of compared this to my estimate, and the intervals overlap. Now I have to say, this is really difficult to calculate, and there's caveats. There's other papers lately one from Mickelson or a very early one from Lee. They're all about this range, but they always give this really big range. And there's problems with the substructure of the macaque and all those things. So since I'm not very experienced in this, I wasn't really sure if I should trust that or be happy that I get the same, kind of a similar result than the population geneticist. So I did something what I normally trust in, likelihood ratio tests, in which I assume that, so the first model is I assume that omega human and omega chimp are the same, and omega macaque is different. And model 2 is a slightly simpler model, where I assume that, again, that omega human and omega chimp are the same. But the macaque is some sort of a multiple of omega human. And so this one is this one's a more general form of this one. So you can do a likelihood ratio test. The difference in degrees of freedom is 1. And I actually figured out that only this is on 10,000 genes, that for 10,496 genes out of the 10,980 I tested, there was no significant deviation observed, which actually means that the differences we see in selection pressures are fairly well described by the differences in population size. So just to sum that up. The population genetic interpretation is I think it's kind of helpful to understand differences between selection pressures, at least if a lot of other factors are very similar, like between human chimp, between the primates. And then for the human chimp macaque tree, the estimates broadly agree with estimates from population genetics, and the differences the likelihood ratio test shows that the differences in selection pressure can be explained by differences in population size. And finally, this was all a lot about reinterpretation. I have another reinterpretation because sometimes I think it's quite inspiring. So this one here is not so interesting, but I think this interpretation is quite interesting for the next couple of days. And for this, I want to thank Siepel and Bustamante, labs in Cornell, Rasmus Nielsen, with whom I had a discussion about this at a meeting in the Newton Institute last year, and Tanja Gesell to discuss this a lot at this meeting. And thanks for listening.
Probably have time for one quick question.